All right, so the digestive system consists of the gastrointestinal tract, which is a continuous tube beginning at the mouth and it ends with at the anus. The main function of the digestive system is going to be to convert food into usable nutrients and energy in your body. There are 7 processes that we will kind of cover as we are going over everything here that are associated with the digestive system. I'm going to go in the order of how the textbook lists them. So ingestion would be the first process, and ingestion simply means eating. Motility, we'll be talking about movement of the contents of the digestive system through the GI tract, and this is going to involve your mixing movements, and we'll actually give specific names to some of these movements depending on where we're talking about these movements and also what kind of muscle is used. And these mixing movements are going to be moving contents of the GI tract back and forth, but kind of just in one place without the movement along the GI tract. And then we'll also see some types of propulsion movements, and these are going to move contents along the GI tract. We will also talk about secretion along the GI tract. Secretion is going to be involving release of the digestive enzymes and also chemicals that are going to aid with digestion of ingested materials. These secretions come from the GI tract, and also we're going to see secretions from the accessory digestive organs. So not just the GI tract parts, but also some accessory organs that we'll be talking about. The digestion of food is a breakdown of food. So we are breaking it down into smaller pieces. We can distinguish between chemical and mechanical digestion. The chemical digestion is going to involve action of enzymes and the actual breaking down of molecules into smaller molecules. Mechanical digestion involves the breaking down of large food particles into smaller ones. So when you're thinking about chewing, you're breaking down larger pieces of food into smaller ones, but you're actually not chemically breaking them down, right? It doesn't involve chemical breakdown of these molecules. So chemical versus mechanical digestion is what we will be looking at. After you digest food, both chemically and mechanically, then into smaller molecules, then the nutrients are going to be absorbed into our blood, and that is known as the absorption. So we're going to move it from the GI tract into our blood. But not everything that we eat is digested. So we are going to have to eliminate whatever is left over, and that's called, the process is known as elimination, and any undigested material will be expelled out of our body in the process known as defecation. So let me click the next slide here. No, I don't want to play. I should have had this slide on, but I don't want to play this for you. So that's basically what I just talked about here. So let's look at our third slide here. The GI, blah, blah, blah. So we can see our structure of the GI tract as well as some accessory digestive organs that we will talk about. So make sure that you know where the GI tract begins. So you're looking at the mouth. That's the first part, and then oral cavity, okay, your pharynx, more specifically, just those parts that are parts of the digestive system, which are the oropharynx and laryngopharynx. Then esophagus, from the esophagus, it's going to lead into stomach. And then from stomach, the contents will enter the small intestine. Small intestine is divided into 3 parts, which we will cover when we actually look at the lower GI tract, although the duodenum, which is the first part of the small intestine, is part of the upper GI tract still. And then from small intestines, the contents will move into large intestine and then exit the body through the anus. So anal canal, and then the opening is called the anus. So that's your GI tract, okay? It's a continuous tube. You could kind of stretch it and it would be just one single tube. Accessory digestive organs you can see on the left side here. So we'll be looking at teeth and tongue, which are in the oral cavity, and they help manipulate and mechanically digest the food. Salivary glands that are going to aid with secretion part of the digestive system. Then liver, gallbladder, and pancreas are also important parts of the accessory structures here. So let's look what's next. And let's do a time to climb. Let's do space today since SpaceX deployed or launched yesterday. So we'll do space. I will hide student names and let's see if you can go the first person. Only one person is gonna play out of E3. Fine, I'll start it for one person, but let's look at the questions. Oh, yay. There you go. All right, good job, everybody. That was close. Good job. So frog won, whoever it was, frog. All right, so let's look at the GI tract wall here. And again, for the exam, you will need to identify, things that belong into upper and lower GI tract. That's just classification types of questions. So make sure that you know what belongs where and what is actually part of the GI tract and what is an accessory structure. Okay, so here we're going to look at the GI tract wall. Again, don't play the sound because I'm telling it to you, but I just realized that I overrode my no sound Nearpod and didn't save the version without the little recordings there. So, but when you do this on your own at home, you can play the sound, which is going to be pretty close again to what I'm saying right now. Okay, so the GI tract wall, here we're looking at a cross-section of the GI tract wall, and specifically this is actually look at the small intestine. And the reason why I know that this is a small intestine is because of those big folds that you can see there inside. But it describes nicely the general structure of this gastrointestinal wall. So it consists of 4 tunics or 4 layers. The innermost tunic is going to be the mucosa, The epithelium of mucosa is going to change based on the region of the GI tract. So depending on where we are in the GI tract, you're going to see different type of epithelium, and that will be kind of important to know what is where as well. That's that. But for the most part, you're going to see that the GI tract is lined by simple columnar epithelium. The areas that must be resistant to abrasions to food are lined by non-keratinized stratified squamous epithelium. And these are going to include the oral cavity, parts of pharynx, and the esophagus, okay? Under the epithelium, you would see the lamina propria, and this is going to hold your small blood vessels, lymphatic vessels, and ends of nerves as well. The last layer of mucosa is muscularis mucosa, and this is the thin layer of muscle that's going to aid in expulsion of secretions from glands into the lumen of the GI tract. So we'll see that when we look at the small intestine specifically, you'll see the structure of the small intestine. You'll see that how the mucosa, the muscularis mucosa, when it contracts, it's basically going to squeeze the gland, and kind of closing it or squeezing it is going to help expel the secretions out into the lumen. Submucosa is mostly areolar and dense connective tissue here, and it's going to house larger vessels and submucosal nerve plexus. We can also find mucosa-associated lymphatic tissue in submucosa and lamina propria of mucosa here. So you're going to find the lymphatic tissue here. But it's also extending into the lamina propria. I didn't mention it when I talked about that part. Muscularis layer of the GI tract is mostly going to contain 2 layers of smooth muscle, and these layers are going to differ in the arrangement of the cells. There is an inner circular and outer longitudinal layer. And we'll see that esophagus and stomach will be a little different here from the rest of the GI tract, but we'll look at that when we get actually to the stomach and the esophagus. The muscularis of the digestive system is responsible for all the movements of the contents of the GI tract. So this is what's going to propel the contents down the line. There are 2 main types of movements: mixing, which is just moving contents back and forth. I've already mentioned that, I think, earlier. And this helps the digestive enzymes act on the food that's being digested, and it speeds up absorption as well. So it's just kind of like turning stuff in place and mixes it with those digestive enzymes. This type of movement is not going to move contents down the line. It's going to stay mainly in one place. So it's just going kind of back and forth. The type of movement that does move things down the line along the GI tract is the peristalsis. So it's a type of propulsion movement. Propulsion means that it's moving along. And this is more like a wave-like contraction of the muscularis. It's going to push the contents along the GI tract. The thickened areas of the inner circular smooth muscle are known as sphincters, and these are going to serve as gates between sections of the GI tract. So every time you hear sphincter, it's a thickened area of this of the inner circular smooth muscle. The last tunic of the 4 tunics is the either adventitia or serosa, and these are only going to differ really in their name, but not in their structure. Adventitia and serosa are both going to contain collagen fibers and elastic fibers, and they're rich in areolar connective tissue, but serosa is going to be completely covered by visceral peritoneum, and adventitia is not. So the only difference between the two is whether they're completely covered by the visceral peritoneum or not. So that's the difference between the two. Okay, so it doesn't say adventitia here on this one. It says serosa there, but again, the difference is whether they're covered by the peritoneum or not. All right, let's see. I've already talked a little bit about motility, but you can see here the mixin types. So think about the mixing as different sections contracting kind of at different times, and that just moves stuff back and forth. Versus peristalsis is more like the contraction begins and then it continues along the line. Okay, let's see if we can do this. Let me see what the pairs are. Okay, yeah. So try to match these. Now, what is what? And I guess we can look at our pairs here. All right, last person needs to make that last match. Good job. All right, so make sure that you know what matches what here. We're now going to look at the regulation of the GI tract. So there are two main types of regulation for the GI tract, as we can see summarized here. The regulation is, or the GI tract is regulated by the nervous system as well as by hormones or endocrine system here. Okay, so both of these can be stimulated by mechanoreceptors or baroreceptors or stretch receptors and chemoreceptors. Okay, so mechanoreceptors are the same as the baroreceptors or stretch receptors. They respond to stretch. Chemoreceptors are going to respond to different chemicals in the area. The gastrointestinal wall houses sensory and motor neurons of the enteric nervous system. This part of the nervous system can function independently of the central nervous system, so make a note of that. Sympathetic and parasympathetic division of the autonomic nervous system also innervates the GI tract. The parasympathetic innervation is going to stimulate the GI tract motility, and sympathetic innervation suppresses motility within the GI tract. Okay, so parasympathetic innervation stimulates motility, sympathetic innervation is going to suppress motility in the GI tract. The nerve reflexes within the GI tract can be initiated by those baroreceptors as a response to stretch and chemoreceptors as a response to changing internal environment. So pH and various types of molecules, so chemicals found in there in the GI tract. We can divide reflexes into short and long reflexes. Short reflexes are local and they're only going to be associated with the enteric nervous system. They have a local effect as well. Long reflexes are associated with your central nervous system, and they affect not only the GI tract, but also secretions from the accessory digestive organs. So they have a little more, like a bigger effect here. For our hormonal control is going to involve 4 hormones. Gastrin, that comes from the stomach. We'll mention it again when we get to stomach. And gastrin is going to increase motility of the stomach. And then you have secretin and cholecystokinin and motilin, and these come from the small intestine. Secretin and cholecystokinin increase secretions from the accessory digestive organs, and motilin initiates motility in the small intestine. So its name kind of suggests what it does, right? Again, 4 hormones. We have gastrin, that is from the stomach. And we have the other 3, secretin, cholecystokinin, and motilin, that's from the small intestine. The secretin and cholecystokinin from small intestine stimulate secretions from accessory glands. motilin stimulates motility in the small intestine. Okay, so I hope that's good there. Okay, so you have them listed here on the slide. We're now going to look at serous membranes that are associated with your digestive system. It's really serous membranes of the abdominal pelvic cavity here. And the serous membrane is known as the peritoneum. This is a double layer membrane. We have a parietal layer that lines the abdominal walls, and then the visceral layer that's going to line the organs. Some organs are completely covered by the visceral peritoneum, and these are known as your intraperitoneal organs. Some organs are not completely covered by the visceral peritoneum because they lie against the posterior abdominal wall. And where they come in contact with the posterior abdominal wall, the visceral peritoneum is not there. It kind of goes through a break. And these organs that are not completely surrounded by this visceral peritoneum are known as the retroperitoneal organs. The mesentery is a term that we use to describe the double layer of peritoneum. So it's just a different name here. And it supports and suspends the organs of the GI tract. What's interesting about the mesentery is that it is full of blood vessels and lymphatic vessels from the digestive organs. So when you look at this figure, you need to be able to tell me which one is which here, the different names of mesenteries here. It's not shown here because this is not an injected specimen, but if we were to do a dissection, you would see in that mesentery a bunch of red and yellow lines, and that would show you all those vessels that are here. You can kind of see them on the mesentery proper on the right side. Okay, you can see how the small intestine, where the membrane is connected to it, has some lines there, and those would be your blood vessels there. Okay, the greater omentum here, this is the one that it's kind of like a fatty. We also know it as a fatty apron, and it's literally like a flap of membrane and full of fat also that covers and protects your intestines. And you can lift it and kind of fold it up. That's why we call it an apron. Okay, so that's our serous membranes. So know which ones they are. There's, I don't know, I think there is an actual regular, like a diagram of these membranes, but I'm not really sure. But we'll see them again whenever we talk about like specific areas of the digestive system. Okay, what are our pairs? And so if you're looking, I should have put the picture there for you as a reference, so you could, be looking at the picture and make those matches there. So if you have a textbook nearby, you can look at that picture again and see which one goes with which. Oh, I didn't. Oops. Why is it showing? Why was it showing the names? All right, good job. All right, let's look at the oral cavity now. And this is a great part to actually study with your own oral cavity, at least for like most of the structures. You can use your own self. So get in front of the mirror, open your mouth, and then try to label everything within your oral cavity that you can see. Some of the structures I want to point out that you might not be familiar with are the fauces. And fauces are the opening of the oropharynx, and it's basically that space. And it's marked by palatoglossal and palatopharyngeal arches. And between these two arches is where you can find your palatine tonsils, if you have them, if they haven't been taken out. If you've ever had like strep throat or sore throat, sometimes you might experience swollen palatine tonsils, and that they're even more pronounced there. But you should be able to see them if you look in the mirror and open wide and try to look in there. If you, something else, if you've ever heard of someone being lip tied or tongue tied, that is involving the frenula, the lingual and labial frenula being too tight, and they restrict movement of the lips and the tongue. This is easiest seen in breastfed babies because their tight frenulum is not allowing them to latch on properly, and they can't get the milk out like they, like they should or enough. And so that's usually it's noticed in babies that are breastfed. The bottle-fed babies, you might miss it because it's easier for the milk to come out of a bottle, so they don't have to work for it as with latching onto the mother's breast. If the lip or tongue tie is not corrected early, then later on it can cause speech problems. So it's corrected later on. So anyway, that's that. But the rest of this, it's really just labeling things. So make sure that you look at this and are able to label what is what here. Okay, hard palate, soft palate. I mean, hard palate is where you have the bone. Soft palate is where you don't see the bone. Uvula is at the end of it. You can see it when you open your mouth. It's hanging down there. That's the end of your soft palate. That's that uvula there. So this is just labeling. Make sure that you are looking at those blank labeling images that are in the Google Drive, and practice that labeling here. Okay. Let's talk a little bit about salivary glands now. Salivary glands produce saliva, and there are 3 extrinsic and multicellular salivary glands, which are shown in this figure. Those are the large salivary glands here. They are extrinsic. We consider them extrinsic because they're not inside of the oral cavity. These are also exocrine glands because they release saliva into ducts, and we know that exocrine glands release things, their products into ducts. We also have intrinsic salivary glands, and these are the unicellular salivary glands that are in the oral cavity. Saliva from these salivary glands is going to have lingual lipase, and this is a lipid digesting enzyme which will get activated once it reaches the stomach. Our salivary glands produce anywhere between 1 to 1.5 liters of saliva per day. And saliva is going to function in moistening your food, and it aids with stimulation of taste receptors as well. Saliva contains salivary amylase, and this is a digestive enzyme that's going to break down starch into simple sugars. You will also find lysosomes and IgA antibodies, which protect the oral cavity from bacterial growth. So that's that. One thing that I would suggest to students to do at home, we would never do that in lab because we don't eat in the lab, but if you have to confirm that salivary amylase indeed does break down starch into simple sugars, you can take a saltine cracker and you can put it on your tongue, kind of not at the tip of the tongue, but a little farther back, and let it dissolve. And initially it doesn't taste like anything because it's a cracker, right? But slowly, in a little bit, it should start tasting a slightly sweet. It should gain like a sweet taste to it. And this is because the salivary amylase is already breaking down that starch in your oral cavity into simple sugars, which we taste as sweet. So you can do that, try it, that it's really the truth and it happens. So that's our salivary glands. Hold on. Actually, before I go into this, I don't want to talk about the teeth yet in a little bit. Okay. But teeth are going to be involved in mastication. Mastication is the process of chewing, and this is mechanical digestion. As far as regulation here, medulla oblongata and the pause control muscles of the lips and cheeks, tongue and jaw to perform the mastication. You have already covered these muscles on your AMP1, so we will not be talking about them here in AMP2, but we will be looking at teeth. So this figure here shows you the structure of the tooth. There are 3 regions of the tooth: crown, neck, and root. And notice the brackets there, okay? If you see a bracket, if you're supposed to label the tooth, if you see a bracket, it's talking about the region. It's either the top part of the crown, neck, or the bottom part of root. The crown is mostly made of enamel, and this is the hardest part of our body, actually, made of calcium phosphate. The bulk of the tooth is made of dentin, and you see the dentin when you look at that figure here. It is present in all 3 parts of the tooth. It's present in the crown, root, neck, and the root as well. The outside of dentin is lined by cementum, and there are periodontal ligaments that attach to the cementum and the bone, and this is what holds the tooth in place. And then inside of the tooth, you have the pulp cavity. This is going to contain a special type of connective tissue that we call pulp. And you'll see, you can see there are blood vessels in there and nerves as well. And these blood vessels and nerves enter the pulp cavity through the root canal. So when you have a root canal done on your tooth, what they do is they basically take out all those blood vessels and nerves from the inside of the tooth. You take that out, they fill it out with something. Sometimes they put rods in there to stabilize the tooth, and then they put a fake, artificial crown on it, on top. And this is better than pulling the infected tooth out and putting an implant, because it is, you're keeping that structure of the tooth in your bone, and implant simply doesn't hold as well as your natural tooth there. So, because there were periodontal ligaments and all that stuff. So that's our tooth. Let's look at how many teeth we have normally, okay? So normally there are 20 deciduous teeth. These are also known as your milk teeth or baby teeth. And then 32 permanent teeth. You don't have to memorize when they come in, but I want you to know which tooth is which, okay? So be able to identify each tooth or the teeth on the picture, and that there's an order to it, okay? So it shouldn't be that difficult there. The first teeth that usually appear in babies are the lower central incisors, then the upper ones, and then the mouth basically falls from the front towards the back, and that's usually the order how they fall out as well. So the milk teeth or the deciduous teeth are replaced by the permanent teeth, and they generally fall out in the same order as they came in, and the permanent ones kind of follow that same pattern. The additional teeth we see in the permanent set are your premolars and third molars. So that's the ones that we didn't have as part of the baby teeth. All right, so, and those times, like where it says months and years, these are all just averages. Everybody is different. I mean, I could see that on my 2 kids. My first one, she got her first tooth like exactly as she turned 6 months, like textbook, right? The little bottom one, right? Central incisor. My second one didn't get her first tooth until she was almost 9 months. And they also started losing them at different times as well. So these are just averages. See, I was holding on to this, thinking that I have time with my firstborn until she starts losing her teeth, and she lost her first one at 4.5. I'm like, I wasn't ready for this. You're growing up. But anyway, so these are just averages. That's how generally they come and go. All right, let's see what I have here. Okay, let's do a little bit more matching stuff as a review. Didn't, why is it showing the names? Sorry. Good, and we just went over this, so it's okay if you don't get it on the first try. It's okay. I don't know if my thing just froze. So remember, you can do in the self-paced one, you can do these again. So I'm going to go ahead and move on to the pharynx and esophagus here. So our food is going to pass from the oral cavity into the oropharynx, and then after oropharynx into laryngopharynx. I want to remind you that these parts of larynx are shared with the respiratory system, and the epithelial lining here is non-keratinized stratified squamous epithelium. So don't forget that, and that's because the food is not quite as mushy as it gets whenever it gets into the stomach. And so it's more solid there. So it needs to be able to, this epithelium needs to be able to withstand those abrasions from the food as it passes through. The pharynx passes food into the esophagus, and then this is a muscular tube that connects the pharynx to stomach. Both layers of muscle of the in the esophagus are skeletal in the upper third. So when you look at the esophagus, the upper third of the esophagus, the muscle is skeletal there. It's still intercircular and outer longitudinal, but it's a skeletal muscle type. And then the middle area is about 50-50 skeletal to smooth. And then the lower part of the esophagus is completely smooth muscle. So it transitions from the skeletal type of muscle into the smooth muscle. The epithelial lining of esophagus is also non-keratinized stratified squamous epithelium. This will rapidly change at the lower esophageal sphincter. And this is where the lining of the stomach begins. Photograph a photomicrograph of this sudden change. You can see this in figure 26.9 in your textbook. So you will see the stratified squamous epithelium. I don't know if I included it, did I? Nope. But you can look at that and you'll see that pretty thick non-keratinized stratified squamous epithelium. That's a bright pink part. And then all of a sudden you are going to see something completely different. So it's not a gradual transition in the epithelium. It's a very sharp, sudden change, and it's where that sphincter is occurring. All right, so let's talk about some processes of the digestive system here. So introduction of the food into the oropharynx is a voluntary process. So we're going to look at deglutition basically. The eating is your first part of deglutition or swallowing. You are going to when you chew, after you chew the food, your tongue is going to move it towards the oropharynx. Okay, so when the food enters the back of your oral cavity into the oropharynx, that's a voluntary process. You're voluntarily pushing it back. When the food is pushed into the oropharynx, this is going to initiate a second part of swallowing, the pharyngeal phase. And this is an involuntary process. So as soon as the food enters the oropharynx, this involuntary phase happens. It involves contraction of the muscles there, and it's going to push the food, which we now call bolus, really, because it's food chewed up and mixed with saliva. It goes into the pharynx, and then the pharynx pushes the bolus into the esophagus. So you have the esophageal phase. That's the third phase. So you have the first phase, the voluntary phase, and this is food is entering the oropharynx. Then you have pharyngeal phase. This is involuntary, and it pushes the food or the bolus towards the esophagus. And then you have the esophageal phase, which is also involuntary, and that pushes the food along the esophagus towards the stomach. It takes about 5 to 8 seconds for the bolus to pass into stomach. When you swallow, your uvula actually kind of closes the opening into your nasopharynx, so the food doesn't go up your nose, but it goes down. And your epiglottis also closes over the trachea, so the food is not going into the trachea, into your or into the larynx, really. So you have those two things that are making sure that food is going where it's supposed to go. It takes about 5 to 8 seconds for the bolus to pass into the stomach from the point of swallowing. Esophagus has to pass through the diaphragm, and the opening in the diaphragm that has to happen, that has to be there, is known as the esophageal hiatus. Okay, so that's our swallowing and esophagus and all that. And this is the esophageal phase. So it just shows you the bolus moving through the esophagus here towards the stomach. Okay, it's moving by peristalsis, by the way. So this is what pushes the food along through down towards the stomach. Okay, so let's look at our stomach here. And stomach is a muscular sac. Its primary function is to contract and mush the food or churn the food into baby food like chyme. So we call this at this point chyme, CHYME. It's not chime, okay, it's chyme. The CH is a K sound. The average amount of food and drink per day is about 3.5 liters, and food stays here anywhere between 2 to 6 hours. It depends really what you ate. The epithelial lining here is your simple columnar epithelium. So we're going to move away from that non-keratinized stratified squamous epithelium to simple columnar epithelium here. Stomach has 4 main regions, as you can see on the image here. The rounder superior part is the fundus. The region where the esophagus connects to the stomach, this is called cardia, and that's why the lower esophageal sphincter, which is located at the border of the esophagus' stomach, is also sometimes known as the cardiac sphincter. So when you hear cardiac sphincter, that's the lower esophageal sphincter. Okay, it's right there. at the cardia of region of the stomach. The main part of the stomach is known as the body, and the narrowed region that you see before the duodenum is the pylorus, ending with your pyloric sphincter. And this is going to regulate passage of contents from stomach into the duodenum. These are places where hold on, no. You can see two curvatures. That's what I was going to say. You can see two curvatures of the stomach. The outer is the greater curvature and the inner is the lesser curvature. These are places where the greater and lesser omentum are going to originate. The empty stomach would have prominent gastric folds, and this allows for stretching of the stomach when the food enters. So you can see the gastric folds on this picture right here. For you. Lastly, we need to talk about the muscularies of the stomach. And I told you in the beginning that esophagus is a little different from the initial picture that we had. And then stomach is also going to have some differences in the muscularies layer. So for esophagus, it was the skeletal and smooth thing. For the stomach, it is actually the fact that there is a third layer of muscularies. So you can still see the outer longitudinal layer. You can see the circular layer underneath that, but the the third inner oblique layer, smooth muscle, is present here as well. And we don't see that in any other area of the digestive system. This different direction of muscle fibers is going to allow your stomach to contract in different directions, in more directions, and it's going to make your stomach more efficient in that churning process. Okay, so adding this different direction of muscle just makes it better contractor, I guess, or better churner of the food. So that's the anatomy of the stomach. Let's talk a little bit about what's happening inside. And this is a cadaver picture of the stomach. So you can see those prominent gastric folds here and such. So, all right, let's look at stomach histology. There are 5 different epithelial cells that you have to be aware of that we can find in the stomach. Also, what's going to be important here is make sure that you know which cells are found in the stomach. Make sure you know which cells are found in the small intestine, because in small intestine, we're going to talk about several different types of cells as well. They're not the same. Okay. So there are, so in the stomach, we have 5 different epithelial cells. 4 of these 5 are secretory cells. So they secrete things into the lumen of the stomach. And one of those cells is going to be enteroendocrine cell. Entero means for gut. Endocrine means that it's releasing hormones. Okay. The secretory cells are able to produce about 3 liters of secretions per day. A lot of stuff that they are able to produce. The cells lining the lumen surface of the stomach are called the surface mucus cells, and they extend a little bit into the gastric pit. So if you're looking at this image here, these are the orange cells here. So those are your surface mucus cells. These cells produce alkaline mucin. Mucin helps to create a relatively thick mucus lining, and this is going to provide kind of a physical protection of those cells from the high acidity that's present in the lumen of the stomach. This mucin is alkaline, so the alkalinity further helps protect those cells by neutralizing the acid at that level. Okay, so that's what the surface mucus cells do. The next, as we're moving deeper into this gastric pit here, you can see the mucus neck cells. So there are mucus cells also, but they're in the neck of the gastric pit. These also produce mucin, but this mucin is not alkaline, it is acidic, and it helps to provide a lower pH environment of the stomach. Even deeper into the gastric pit, you can see parietal cells, chief cells, and G cells. Parietal cells produce substance known as intrinsic factor, and this is necessary for proper absorption of B12 in our small intestine. And B12 is necessary for normal production of erythrocytes. So this function of the parietal cells is extremely important in our body. Okay. Another function of parietal cells is aid in formation of hydrochloric acid. Parietal cells produce hydrogen ion by splitting a water molecule into hydrogen and hydroxide ion, and then this hydrogen is pumped out into the gastric pit, and hydroxide will combine with carbon dioxide that produces bicarbonate ion, and that is moved out of the blood. Hold on, do I have a picture of this here? Yes, right here. Okay. As the bicarbonate ion is moving out of the blood, out of the cell into the blood, it is exchanged with chloride ion, and then chloride ion is pumped by the cell into the gastric pit. So what's happening in your gastric pit is that the hydrogen that was expelled and the chloride ion that was expelled meet in the gastric pit, and this is where they form the hydrochloric acid. So technically, the parietal cell is not creating hydrochloric acid, it's just sending components of the hydrochloric acid into the gastric pit, and that's where that acid forms. You don't really want to have acid inside of the cell. It would start damaging the cell from inside, and that's the reason for that. Hydrochloric acid is important for several things. The hydrochloric acid lowers the pH to about 2, and making this, so this makes and maintains the high acidity in the stomach. This process begins the chemical digestion of proteins, because low pH is going to denature or unfold the proteins. And in order to denature protein, you have to break some bonds that hold that protein in its shape, right? So it starts the denaturation process of proteins. Hydrochloric acid also activates pepsinogen molecules into active pepsin. And in general, it helps break down plant and animal matter that you have ingested. So that's your hydrochloric acid. Going back to our cells here, let's talk about some more of these cells. Next on our list will be the chief cells. Chief cells produce pepsinogen, and pepsinogen is an inactive protein digesting enzyme. It is important that this enzyme is inactive inside of the cell, because if it was active in the cell, it would start digesting proteins inside of the cell. Okay, so all your protein digesting enzymes will be inactive until they reach the lumen of the GI tract. Okay, so not just pepsinogen, but all the digestive enzymes that are protein digesting enzymes are inactive until they're activated in the GI tract. Okay, so the pepsinogen gets activated by in the stomach lumen, either by pepsin molecules that are already active, or it can be activated by hydrochloric acid. This, it depends on what molecule this pepsinogen meets first. Okay, so if it meets hydrochloric acid, then the hydrochloric acid activates it. If it meets pepsin, then the pepsin activates it. So that's pepsinogen. Chief cells also produce gastric lipase, which along with lingual lipase from the intrinsic salivary glands will digest small amount of ingested lipid. Lipase for digestion of lipids. Okay, so lipase digests lipids. That one should be easy to tell. The last types of cells is going to be the G cells. And the G cells are those endocrine cells. These are not cells that produce secretions. These are cells that produce hormone. They're endocrine cells. And this hormone is known as gastrin, and gastrin goes into the blood. The hormone will increase motility of the stomach and secretions of the other cell types. Okay, so that's what gastrin does. But it's a really big player in motility of the stomach. So that's that. Okay, last thing that I will talk about today is the regulation of the digestive processes here. So the stomach is going to contain these cells that are known as pacemaker cells, and they're responsible for the contraction rhythm of the stomach. This is about 4 contractions per minute. So the stomach contracts about 4 times each minute. These contractions remain constant in their rate. So the contractions don't increase in their frequency, but the strength of the contraction is what changes depending on various stimuli that are received by your GI tract. The events of the stomach and digestive system can be divided into 3 phases. We can talk about cephalic phase, gastric phase, and intestinal phase, okay? Cephalic phase is activated by thought, sight, or smell. So just think about when you are hungry, we're getting close to lunch here. So if you're hungry, you can see some picture of some food, or you just think about some food, right? You smell something really good. You can notice that your salivary secretions increase, right? Your stomach might have started doing things. That's your cephalic phase, okay? Cephalic phase includes or involves increase in the stomach's motility, increase in the strength of those contraction, and it also is going to increase secretions from gastric glands as well. The second phase is your gastric phase, and gastric phase is initiated by presence of food in the stomach. So after you eat and as the food is entering your stomach, gastric phase is happening. This is causing distension of the stomach, right? After you eat a meal, your stomach will stretch, okay? That's distension. And this is also going to change pH inside of the stomach because you're mixing food that you just ingested with the acid in there. And these are going to be signals to increase stomach's motility, saying, hey, there's something in the stomach, we gotta churn stronger, okay? And work on the mechanical and chemical digestion of the contents here. This also releases gastrin. So distension causes release of gastrin as well. Now there's food in the stomach, so stomach must churn and mix it, right? And after this is the intestinal reflux. Intestinal reflux is initiated by acidic chyme in the duodenum. So as the chyme is leaving the stomach, it's acidic, right? And as it enters the intestine, this is going to initiate this intestinal reflux. So this means that food has been churned already. It is leaving the stomach and it's entering small intestine. So since the volume of chyme decreases, the motility of the stomach will decrease as well. So stuff is leaving your stomach, and so you don't have to churn it as strongly anymore. Cholecystokinin and secretin are going to be released from the small intestine as a response to chyme. The presence of lipids in the chyme is going to stimulate cholecystokinin, and high acidity stimulates secretin. But both of these are going to increase secretions from liver, pancreas, and the gallbladder. And that will make a little more sense why this is true when we actually look at the liver, pancreas, and gallbladder and their secretions. It will nicely fall in place why both of these hormones increase these secretions based on these stimuli. So that was the last thing that I wanted to talk to you about today. And we're going to, your digestive system part 2 is going to cover the lower GI tract, and there's actually going to be part 3 as well, because the audio makes the file really big, and so I had to split that into 3 parts, but the third part is short. So yeah, so digestive system it's a lot of stuff, right? But hopefully not too difficult. Work on those figures, work on your labeling, and then know how to classify the different things, be able to match the things like we did in the practice for the Nearpod, and you should be good. Know which cell does what. Definitely know which cell does what. That's important.